morning students. Today we are going to start next chapter that is about enzymes. First we need to understand. It is very familiar term for all students. What is enzymes? While studying most of the reactions and don't need to go any other topic. Just before in the last chapter also we have come across so many enzymes. So regarding that enzymes, we are going to learn in this chapter. Enzymes are biological catalysts produced by living cells. What is its function? It increases the rate of biochemical reactions without being used up in the process. There is a definition. What are enzymes? Enzymes are biological catalysts produced by living cells which increase the rate of biochemical reactions without being used up in the process. Next, we are going to see some of the properties of enzymes. Intact structure. All enzymes should have an intact structure. That intactness is very necessary for its function. If any disturbances or distortion happen to the shape, that enzymes then cannot mediate that particular reaction. So, its structure should be stay intact. Then endoenzyme. Endoenzyme. What are endoenzyme? Enzymes that function within the cell are called endoenzyme. Example you should give. One example is cytochrome oxidase. It's a familiar enzyme for you. Cytochrome oxidase. While learning about that biological oxidation also, we come across this enzyme. Then exoenzyme. Endo means function within the cell. Then exoenzyme means enzyme that functions outside the cell. Then what is inducible enzymes? Enzymes form in the presence of its substrates only. That types of enzymes are called inducible. The presence of substrate induce the formation of that enzymes. Example is lactate dehydrogenase. Then what is constitutive enzymes? Constitutive enzymes are produced, they are produced independent of the presence of substrate. Even if a substrate present or absent, that doesn't matter for the formation of such type of enzymes. It is produced independent of the presence of substrate. Example is cytochrome oxidase. Cytochrome oxidase familiar. Then cymases. Cymases. What is cymases? They are called active enzymes. These enzymes, they didn't need any modification for acting on substrates. These enzymes didn't need any type of modification to act on substrate. Example is glycolytic enzymes. Then zymogens. Zymogens are otherwise called proenzymes. These enzymes are actually produced in inactive states. Later on only they become active. Such type of enzymes also we have learned in our first chapter in physiology. That example come here also. They are trypsinogen, pepsinogen. They are all digestive enzymes that help in digestion. Then simple enzymes and conjugated enzymes. Simple enzymes means these enzymes are formed only of proteins, no other compounds. So on hydrolysis that yield only amino acids. Why it yield only amino acids? Because it is composed of only proteins. Proteins is composed of amino acids. Examples are amylase, urease. Then what about conjugated enzyme? Conjugated enzymes, these enzymes have a non-protein part and a protein part. And that protein part have a name called apoenzyme and non-protein part another name called cofactor. So conjugated enzymes always composed of apoenzyme and cofactor. Apoenzyme is the protein part of that conjugated enzyme and the cofactor is the non-protein part of that conjugated enzyme. Then both these apoenzyme and cofactor are necessary for the catalytic action. and these both together form the hollow enzyme. When these cofactor and apoenzymes are present together, then that enzyme is called a hollow enzyme. Then one more point. These cofactors are of two types. Two type means they are prosthetic group and coenzyme. What is prosthetic group? Prosthetic group means those cofactor that is firmly bound to the apoenzyme. If any cofactor, if it is firmly, that word we need to give attention. Firmly bound to the apoenzyme. Such type of that cofactors are called prosthetic group. Then what about the other one that is loosely bound? That become coenzyme. Coenzyme is another type of cofactor in which that cofactor binds very loosely to the apoenzyme. Examples for such type cofactors are NAD, NADP. Okay, next we are going to see the properties of enzymes. Properties of enzyme, first property is a catalytic efficiency. The enzymes have a catalytic efficiency by which it can increase the speed of a reaction to 10 raised to 3 or 10 raised to 8 times than that of an uncatalyzed reaction. And also one more term we need to familiarize, that is turnover number. Turn, turnover number. What is turnover number? Turnover number is nothing. It is the number of substrate molecule converted to product by one enzyme molecule per second. So how in the form of a definition we can tell that yeah. Turnover number is the number of substrate molecule converted to product by one enzyme molecule in 1 second. That is turnover number. And what determines the efficiency of the enzyme? The ability of that enzyme to bring down the activation energy very fast. That type of enzymes are more efficient. Then as we are going down the topic, we will learn about the activation. What is activation energy? Now just hear that. Those enzymes which you can very fast bring down the activation energy, that type of enzymes are very efficient. So next term is active site. Active site means these enzymes contain certain sites. These sites are called active sites. What is this peculiarity? That site actually that substrate are binding. And so that site should contain some specific sequence of amino acids. And also that site should be complementary to the substrate with respect to its shape. These are the properties of active site. Then specificity. The enzyme should be highly specific. And while talking about specificity, we need to understand that there is two types of specificity. Which are the two types of specificity? One is substrate specificity. One is substrate specificity. That means that enzyme act only a specific substrate. Second specificity is called reaction specificity. Reaction specificity means that enzyme will catalyzes only a particular type of reaction involving a substrate. Actually, that substrate can undergo so many reactions, but that enzyme catalyzes only a particular reaction involving that substrate. That is one another type of specificity. That specificity is called the reaction specificity. Example, urease. It shows this both type of specificity, that is substrate specificity and reaction specificity. Then regulation of activity. How can we regulate? So many substances are present. A number of substances are present. They all can regulate the activity of enzyme based upon the need of the cell by activating or inhibiting the enzyme. Example, phosphorylase can be activated by calcium ions. Then sensitivity to heat. Enzymes are highly sensitive to heat. If the temperature is above 60 degrees Celsius, then permanent denaturation will be the result that enzyme will face. Enzyme will completely denaturate when the temperature rises above 60 degrees Celsius. Low molecular weight enzymes are more stable than high molecular weight enzymes. Then reversibility. Reversibility means most of the enzymatic reactions are reversible. It can proceed forward and backward direction. Next, we are going to see the mechanism of enzyme action. In the mechanism of enzyme action, we need to familiar with so many terms. That is, first one is enzyme substrate complex. Whenever an enzyme interact with the substrate, first it forms a complex. That complex is called the enzyme substrate complex. This is a key part of the enzyme action. And the enzyme increase the rate of reaction by lowering the energy of activation. That is activation energy. For all reactions, there will be an activation energy. By lowering that activation energy, only that reaction proceed very fast. Then we need to understand activation energy. Only on reaching a certain energy level, that reaction proceed forward. So if that activation energy is very high, that the reaction will not proceed quickly. So we have to means that enzyme have to lower that activation energy to proceed that reaction in forward direction very fast. For increasing the speed of that reaction, what the enzyme need to do? The enzyme need to lower the activation energy. And for all reaction, there will be an activation energy. If any reaction which has high activation energy, that reactions rate will be very slow. Then came an enzyme. That enzyme lower that activation energy and speed up that reaction. For understanding that, if those who clearly understand this thing don't need to listen to the example that I am going to say. Others can listen to the example. A simple example. Example means suppose there is a heavy table in front of you, and for moving or pushing that table, anybody need the x amount of energy. And if one person is coming and try to push that, if that person didn't have that x amount of energy, that table will not move. Then think that one more person and giving a hand to push this table. Then the first person need only very less energy to move that table. That means the initial required amount of energy is lowered with the help of that second person. In that situation, you need to replace that required amount of energy x, a value I already tell you in the beginning. That value you need to replace with the activation energy. Then you think by your own way. It will be clear. Activation energy is only this much. For all reaction to proceed forward, certain amount of energy is there. If whatever reaction which need higher activation energy, it will always proceed very slow. For such reaction, if an enzyme is present, that enzyme lowers that activation energy, and by lowering that activation energy, it helps that reaction to proceed forward very fast. Then I need to understand about ground state and transition state. Ground state means it is the beginning point of a reaction. In the starting point of a reaction, that state is called the ground state. The transition state is simple. It is that state. In that state, that substrate forms a complex with the enzyme. After forming that complex, it's very easy for the substrate to get breakdown into products. That is the transition state. Activation energy, but I very clearly explained to you, it is the energy barrier that separates the reactants from the products. Okay, it is a type of energy barrier itself, which separates the reactants from products. That barrier actually separating the reactants to become products. Higher the activation energy, lower will be the reaction rate. How we can lower this activation energy? When we are going to understand how that activation energy is lowered by the presence of enzyme, that we are going to understand. Whenever a substrate is bind with a enzyme, that binding is actually through some weak bonds like a hydrogen bond, van der Waals force, ionic bonds, hydrophobic bonds, or hydrophilic bonds. Through these bonds, these type of bonds, the substrate is binding with the enzyme, and this binding release a little amount of energy. The name of that energy is called binding energy. And this energy actually opposes the activation energy. And this opposition helps to lower the activation energy. Understand? That binding energy opposes the activation energy. And in that way, raise the reactant molecules to its transition state. In the transition state, its activation energy is very less. So it can very easily convert it into products. In that way, enzymes help to lower the activation energy. Then we are going to see Michaelis-Menten enzyme substrate complex theory is very, very important. This theory is telling that enzyme first bind with the substrate reversibly. Reversibly means it can bind and also unbound with the substrate. And after binding with the substrate, forms a complex called the enzyme substrate complex. After forming enzyme substrate complex, what is forming? Products are forming. And with the release of products, uh, the products, what is freeing? That enzyme is again become free for starting another round of reaction. That is Michaelis-Menten enzyme substrate complex theory. It's very simple. And regarding the active site of enzyme, that active site is actually created by the coiling of tertiary structure. Primary structure, secondary structure, and tertiary structure. I have in very detail I explained to you in the biomolecules chapter. So, if anybody have any doubt, you go back and see what is tertiary structure. By coiling of the tertiary structure, these active sites are creating. And these active sites are usually in the form of some cleft or crevice. Crevice means വിള്ളൽ പോലൊക്കെ ഒരു കുഴി പോണം അങ്ങനെ ആയിരിക്കും ഇരിക്കുക. And this active sites actually determines the specificity of enzyme action. Next, we are going to see models for the formation of enzyme substrate complex. Very, 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 very important. Okay, it is asked for several times for university examination. Lock and key model, very, very important, and it's very easy. It is proposed by Emil Fischer, 1894. What is telling structure of the active site is complementary to the structure of substrate. That word we need to give more stress. Complementary. What is complementary? The structure of active site is complementary to the substrate. Like a key fits to a lock. That's why this model is called a lock and key. And what is the limitation of this model? That rigidity of the active site. As the active site's shape is highly rigid, it will not permit any variation for the features of substrate. If any variation happened to the substrate feature, that substrate could not come and bind with that enzyme. That's a limitation. And also, this theory did not explain the flexibility shown by the enzyme. Normally almost all enzymes show such flexibility. What flexibility? In the presence of allosteric modulators, uh, it shows some flexibility. Flexibility means concerned with the rigidity of the active site, rigidity of the shape of the active site. Normally, the enzyme shows some flexibility. It shows some, it is not much firm with regard to that active site normally in normal situation. But according to this law, that structure, the shape of the active site is very rigid. It should not be changed for binding with the substrate. Next model is induced fit or a Koshland model. That is proposed by Koshland in 1963, and it explained the flexibility of the active site. It explains that active site is not rigid, but what type? When a substrate command bind with the enzyme, its active site undergoes a conformational change. That means the orientation of the pattern of that coiling undergoes some change that occurs when after the substrate come and bind with the active site. So in that way, according to this model, that binding of substrate to the enzyme is a two-step process. First step is the initial or primary binding. In that binding, substrate binding induces a conformational change to bring the correct orientation of substrate with the enzyme for bringing that correct orientation. And secondary or final binding occurs after that only, after the initial binding. And the secondary or final binding is the most fitting binding. Okay. That's about the two models for the formation of enzyme substrate complex. Next, we are going to see the mechanism of bond formation, how the bond is formed, and also how breakage of bond is occurring by the catalytic functional group. So many functional groups are present in the active site of enzymes. That we are going to see how the bond is forming between substrate and the enzyme, how the bond is breaking between substrate and the enzyme with the help of catalytic functional group that present at active site of enzymes. First method is acid-base catalysis. This method is usually used to avoid formation of secondary reactants, And the secondary reactants means unstable intermediates to prevent the formation of unstable charged intermediates in a reaction. This method is usually used. Then we are going to see what's the method. This is the enzyme, this is the substrate, and they are coming to bind with the enzyme. What is happening then? Here proton is transferred from enzyme to substrate, or proton is exchanged from substrate to enzyme. Through that exchange, what is forming? An enzyme substrate complex, a particular compound is forming, and that compound is easily breakable into products than any type of a secondary reactants or intermediates. This is one method that used to prevent the formation of any intermediates in the reaction by the exchange of protons, transfer of protons between substrate and enzyme, forming a compound. That compound is easily breakable into products. One method. And uh, For example, amino acid like glutamic acid and aspartic acid, the arginine can, then we are going to understand who is donating that protons and who is accepting the protons. Consider, suppose this is the active site of enzyme. As we know, active site contains some specific amino acid. Each amino acid has some functional group. That functional group acts as proton donors, or at the same time sometimes acts as proton acceptors based upon whether they are acids or bases. Okay, amino acids like glutamic acid and aspartic acid, arginine, these amino acids can act both as acid and base. That means they can either donate or can accept proton. Whatever it be, this acceptance and donation helps to the formation of this compound that can be easily breakable to form products. Example for this acid-base catalase is given by the action of ribonuclease. This ribonuclease act by acid-base catalysis method to avoid the formation of intermediates in the reaction. Next, substrate strain. Example, this action of lysozyme. When a substrate binds an enzyme, it induces a strain in the substrate. And that strain increases the reaction rate. That's called substrate strain that we are going to see as the enzyme. And these enzymes active site contain specific amino acids like glutamic acid and aspartic acid. And this is the mucopolysaccharide that is present on the bacterial cell wall. This enzyme need to break this bacterial cell wall. How it break this bacterial cell wall? Here, that glutamic acid and aspartate present in the active site of this enzyme act on the glycosidic bond present between the fourth and fifth monosaccharide units of the mucopolysaccharides and hydrolyze it. In this way, it breaks the mucopolysaccharide found in the bacterial cell wall. Next is a covalent catalysis. Covalent catalysis means enzyme form a transient covalent bond with the substrate and result in product generation. That is covalent catalysis. Example, enzymes with serine residues at the active site mediate the reaction by this method, covalent catalysis. Then metal ion catalysis. That is it. Metal ion catalysis means some enzymes need the presence of certain metals to facilitate the easy formation, the speedy formation of products. These ions actually help to establish weak bond with the substrate. What is the role of that ions here? That ions actually help this enzyme to establish weak bonds with the substrate to facilitate the fast conversion of substrate into products. Example, ferrous and ferric ions. With this, we can finish today's section. Thank you.